There's a chart that's at the end of your chapter which shows the differences between the three types of muscle tissue, and it's very important for you to study from it. So, for the body location for skeletal muscle, we know that it's attached to bones. Cardiac muscle is in the walls of the heart, and smooth muscle is in hollow organs like eye muscles, airways for respiration, large arteries. We also find it in other blood vessels as well. In the GI tract, here we can see the stomach. It's in the respiratory system, and it's also in the skin in a muscle called the erector pili muscle. So the goosebump muscle. The second row is the cell shape and appearance, and these will definitely be on your practical images like these. So I would recommend using a tool in the study area of mastering A&P, which is called Practice Anatomy Lab. And hopefully you've already used that for the histology part of your lab. Again, there's no images on the lecture exam, but there will be images on your lab exams. So, the shape of the skeletal muscle is very organized. They are long, cylindrical, multinucleate with these striations, these stripes. Cardiac muscle is also striated, but it's branching chains of cells and usually has just one or two nuclei. Remember, it does have these what are called intercalated discs that are present in cardiac muscle. And smooth muscle is non-striated and it's spindle shaped and uninucleate. It sort of tapers off at the end of the cell. The connective tissue components are shown in the next row, and we learn for skeletal muscle that it has epimysium, perimysium, and endomysium. The um, cardiac muscle also has endomysium around its individual cells, and there's endomysium in smooth muscle as well. For our next row, we have the presence of myofibrils, which are only in skeletal and cardiac muscle, but not in smooth muscle. There's actin and myosin filaments and dense bodies which anchor those actin filaments. The next row shows the presence and location of T tubules. They are, of course, in the skeletal muscle. We see the T-tubule here, the sarcoplasmic reticulum, and the organization of the sarcomere. There are sarcomeres that are in cardiac muscle as well, and one T-tubule per sarcomere at the Z disc. So not as many as in skeletal muscle. And then in smooth muscle, as you just learned, there are no T-tubules. Instead, there are caveoli. And remember, there is no neuromuscular junction, and the calcium in smooth muscle binds to calmodulin, not troponin. And maybe you can remember that more easily because it starts with Ca, like calcium. When we look at the rest of that chart, we can see it here. The sarcoplasmic reticulum is very elaborate in skeletal muscle. There's not as much in cardiac because a lot of the calcium comes from the extracellular fluid. And smooth muscle is very similar as well because a lot of that calcium also comes from the extracellular fluid. There is no gap junctions in skeletal muscle, but there is in cardiac and smooth. And those gap junctions in cardiac are in the intercalated disc. The intercalated disc is only in cardiac and not in skeletal or smooth. The cells do exhibit a neuromuscular junction for skeletal, but not in cardiac or smooth. As far as the regulation of contraction, they're voluntary in skeletal muscle via the somatic nervous system, but involuntary in both cardiac and smooth. In this case, the cardiac, they're controlled by the autonomic nervous system, hormones, and stretch, and autonomic nerves also control smooth muscle. The source of calcium is from the sarcoplasmic reticulum and skeletal muscle, but in cardiac and smooth, it's from the extracellular fluid in addition to the sarcoplasmic reticulum. Calcium regulation. Troponin is on the actin containing thin filaments. Troponin is also on the cardiac muscle, actin containing filaments. This is one of the reasons that in the hospital, they'll oftentimes check the blood for troponin levels because that could indicate a heart attack. It doesn't always, but a lot of times it does. But remember, for smooth muscle, instead of troponin, we have calmodulin that the calcium binds to. There's no pacemaker in skeletal muscle, but in both cardiac and smooth, we have a pacemaker because it's controlled by the autonomic nervous system. The nervous system stimulates or excites the skeletal muscle, but it can excite or inhibit both the cardiac or smooth muscle. As far as the speed of contraction, skeletal muscle can vary based on things like recruitment, the action potentials, various factors that you've learned. However, in cardiac muscle, it is slow. And this may seem kind of weird, but it maintains a very specific heart rate. It obviously can increase and decrease from other factors. And smooth muscle is the slowest. It's very slow. Think about how long it takes foodstuffs to go all the way through your digestive system, through your small intestine and then large intestine. The skeletal muscle doesn't really have any rhythm, but cardiac and smooth definitely do. The skeletal muscle, the strength increases with the degree of stretch to a point, and the same with cardiac muscle. However, smooth muscle has what's called stress relaxation response. As far as metabolism, we've learned that skeletal muscle can contract both aerobically and anaerobically. Just to remind you, the aerobic is slow oxidative, but anaerobic relies on glycogen and it's fast twitch glycolytic. The other one in the middle of that, you don't have to know. Cardiac is only going to contract or get ATP aerobically, and smooth muscle is mostly or mainly aerobic. So the mechanism of action for contraction for smooth muscle, you don't have to know it in as much detail as you learn for skeletal muscle, besides some of the generalities which are shown here. So just like the last table just showed you, smooth muscle is slow, synchronized contractions. It's electrically coupled by gap junctions. It's self-excitatory because it has the pacemakers like cardiac muscle does. It has actin and myosin still that interact by the sliding filament mechanism. The final trigger is increased intracellular calcium level. And remember, that's from the extracellular fluid as well as the sarcoplasmic reticulum. The contraction of smooth muscle is different from skeletal muscle by some of the ways that you already know. Calcium is still obtained from the SR, but mostly from the extracellular space, from the ECF. Calcium binds to calmodulin instead of troponin, and there are cross bridges with actin. Stopping a smooth muscle contraction requires more steps in skeletal muscle. For example, relaxation requires the calcium to detach from calmodulin. And like skeletal muscle, there's also active transport of calcium into the SR, but also the extracellular fluid. The energy efficiency of smooth muscle means that it's slower to contract and relax, but maintains contraction for prolonged periods with little energy cost. So this is because it has a slower ATPase, which remember is still the myosin. So that's really good because, for example, the colon can maintain this contraction for a long period of time. Smooth muscle maintains a moderate degree of contraction constantly without fatiguing, and this is referred to as smooth muscle tone. And it makes ATP mostly via aerobic pathways. The regulation of control is somewhat like cardiac. It's by nerves, hormones, and even local chemical changes. Like there could be irritation on the inside of the GI tract, and that causes it to change. And the neuroregulation is still a neurotransmitter binds, causes either a local graded potential or an action potential. And you'll be learning more about the graded versus action potential in chapter 11. But the result is still about the same. It increases calcium concentration in the sarcoplasm, and the response depends on that neurotransmitter released and the type of receptor. So one neurotransmitter could be stimulatory or inhibitory on a different organ. So one really good example of this is when we think about the bronchioles, which have smooth muscle in them. As you probably know, adrenaline, which is epinephrine, is used if somebody has an asthma attack, for example. That's the active ingredient in albuterol. So that is going to cause dilation of the bronchiole. So that would be the example of the neurotransmitter. Whereas the neurotransmitter acetylcholine is going to cause constriction of the bronchiole. And this is really interesting because it's how different medicines work. And you'll learn all about that in chapter 11 as well.